Today I'm going to talk about the principles of fluorescence or fluctuation correlation spectroscopy. This is a technique that you might want to use in your research group. It's going to tell you a lot about how proteins move, how they diffuse, how molecules diffuse. So yesterday you heard from Professor Dave Jamieson about QVET studies, and we're going now from, let's say, some QVET, which you can actually get a lot of information from measuring things when they're purified in a beautiful solvent, to more of the QVET, and we can do QVET assays using fluorescence correlation spectroscopy into the or you can also do it in the microscope, where we're looking at molecules diffusing in living cells. So Robert Brown was a botanist, and his picture that I'm showing over here. So in 1827, the botanist Brown noticed that pollen grains, this is what you could, sometimes you go outside in the field and you notice there's pollen on you. This is what gives you a lot of allergies. They actually moved, they zigzagged, right? And they jiggled under the lens of the microscope, and he noticed that this was interesting. And back in the day, in his time, he was thinking that perhaps this movement explains something known as the germination theory. So how do things come to life? He had a theory where he proposed that every living system actually moves, right? So if you observe it under some sort of system, it could be a microscope, it could be like a camera, that things move. And the funny thing is that he took it up to the Royal Society in England, and he says, everything that is living moves. And they said to him, you have to prove this, right? How do we know that inanimate objects don't move? So he said, okay, I'm going to take some wood grains from, some wood is surely dead, it's no longer living. It used to be alive, but they're no longer living. So he's going to watch it. And indeed, those wood grains moved. All right, he said, okay, well, let me test something else. It was alive, maybe something that was living. I'm going to take some really, really old wood. So I'm going to go to the London Museum in London, and I'm going to scrape some wood from these sarcophagus that they got from Egypt. So he did that. He's like, surely these are dead. They've been underground forever. They're not going to move. Indeed, those moved, right? So his theory was put to the test, and it failed. But it wasn't until 1887 that found that the Brownian motion was more than a rapid, where it was more rapid for smaller particles. So that was really important because the size mattered. How big a molecule is will depend on its diffusion coefficient. We're going to talk about that when we talk about FCS. And, it wasn't actually until Albert Einstein in 1905 explained why particles moved, right? He did it with a mathematical equation deriving the kinetic theory of gases that was developed by Boltzmann and Gibbs, that it explained the randomness of the motion. So how can we measure the molecular exploration of space and time? Here you're looking at exactly what Brownian saw under his microscope, where these are particles that are just randomly diffusing, randomly moving, right? We can actually label and tag fluorescent molecules. What does it look like on our microscope? It looks like this, pretty much noise. But believe it or not, the fluctuations are stored within this image. So in a crowded environment with dim molecules, we can actually measure single molecular detection of molecular movement. And we're going to talk about how we do that today. So fluctuation dissipation principle explains that if we perturb a system from, let's say, just the normal non-perturbed system, if we actually perturb it in some way, the explanation is that you're going to have a high amplitude because they're perturbed, right? And eventually those molecules will come back to steady state. So this can happen if we add energy to the system, but it can also happen spontaneously, right? So this is spontaneous fluctuation, and then as a function of time, they will return back to steady state. And so this fluctuation is in a part of a system can cause a system to locally go out of the equilibrium, and these spontaneous fluctuations dissipate at the same time constant as if we had externally perturbed the equilibrium into a system. And this is an important concept because we can actually measure how long it takes for that system to come back down to steady state. All right, so imagine that we take some beads and we collect the beads and we hold them, right? So this is what you're seeing in the center. And Fick's law and the diffusion, let's say, principle states that as a function of time, if you let go, right, as a function of time, these particles will move away from the center. So what you're seeing is if we're observing them at time 1 or time 0, we see that the molecules are in the center, and as time progresses, those molecules will move away from the center. And Fick's law states that this is the probability distribution of how molecules will be found later in time at a given distance, right? So we can actually calculate how long those molecules will take to go to a given distance because of their movement and time. So we can measure the how fast the substance diffuses, which is directly tied to both the concentration gradient and diffusion coefficient. So how fast those molecules move. Now, typically, scientists and biologists can use another type of method to measure this. They can use something called fluorescence recovery after photobleaching. And you can use a microscope, and here's the actual idea behind this. And this was developed by Dan Axelrod, who I'm showing in this picture. And as a function of time, if we bleach a given spot, right, completely destroying the molecule, bleaching means there's no those molecules are gone. They're no longer going to emit any photons. So if we bleach a particular spot, what will happen, and this is perturbation, right? We're perturbing the system, is the intensity will go down because they're going to be dark. And then as a function of time, as molecules move in, we're going to get a recovery of intensity from those molecules that are repopulating that space. And we can now calculate the time of recovery that gives us, say, the average time those molecules took to come back into that spot. Now, this is a great method because you can learn a lot about your system. You can learn if something is moving really fast. You can learn if something is static and it's stuck and that you don't get any recovery. And you can get that information. However, with this system, you're measuring a bulk rate, right? We're not looking at single molecules. We're not counting how many molecules are in that spot, or we don't get that information. But with another type of analysis, instead of bleaching that spot, if we simply just stayed in one area and not bleach the molecules, we have low intensity, we can now monitor, detect the molecules as they go in and out of that spot. And these are the fluctuations that we see. And a little bit later, I'm going to talk about how we get these fluctuations. How is it that we detect these fluctuations, right? And now you can use a mathematical calculation. We say, okay, is this fluctuation from the same molecule? And the property of FCS is that you have to be low concentration and that molecule has to be the same for it to correlate as a function of time. So we see this decay, okay? And I'm going to talk a little bit more about this. All right, so this is the fluctuation. What we're measuring is fluctuation of intensities, and we can use the correlation spectroscopy to get that value, the number, the diffusion time. And we can also get the number of molecules. How many molecules were there, right? Okay, so to help you understand the number, let's go back to 1911 to Schwetzberg and Inoue. And there's these two colleagues, and Professor Schwetzberg told his trusty assistant, Dr. Inoue, okay, What we're going to do is we're going to take the newest microscope back then, 1911, which was this, forgot exactly what it was called, but it was a microscope that allowed them to look at single colloids, gold, let's say, particles that were made out of some metal. And what you see here is their actual data where they're counting the number of molecules. So at some point, Dr. Schwetzberg, he had a metronome. If anybody knows what a metronome is, a clock. So he said, every second, I'm going to tell you number of molecules I see at that time. Okay, so he saw 1, then he saw 2, then he had 0, 0, and he kept counting. And eventually his eyes got tired. He had to blink. There's a gap, right? Okay, no data. He continued on. And so this is just collection by visual inspection, right? It's a similar idea. Now we take that data, we use our modern, let's say, graphing capabilities, and we can plot now the number of particles, the same ones that you saw before, but now this more fancy way to do it. And what do you see here is that if we now count the number of particles and calculate the frequency of those numbers, right? So at time 0, or let's say at the number of particles that we saw 0 times were most of the time, right? The frequency of the number of times we saw 0 particles, the number of times that we saw 1 molecule, 2 molecules, and so forth. So this behavior tells us, this shape of this distribution tells us that it's Poissonian, right? So this type of behavior, the diffusion and number related to the number of molecules, has a Poissonian behavior. So this is valuable information because from the fluctuation analysis, we can actually obtain the number of molecules that we see. And I'm going to show you how we did this for the autocorrelation function and how this is calculated. All right, so now back to modern days, no longer in 1900s. How do we do this in a microscope? In a microscope, we have these optics that allows us to focus the laser beam in a very small focus spot. This is called the volume of the point spread function, or PSF. There are many terms for it. So this observation volume enables us to both calculate the volume of that point spread function, whether we're using 1 photon or 2 photon excitation, we have to calculate like geometry of this volume. And as molecules diffuse in and out, they're going to give off a photon burst, right? So the molecule is not going to be emitting photons the entire time. It's only going to emit when it's excited. So the molecule will be excited, it'll give off a photon burst. It'll continue to roam around, it'll give another photon burst. These are the fluctuations that we're observing. And based on those fluctuations, we can, what is observed? We can get the rate of motion, which is diffusion. We can get concentration of molecules, and we can get changes of the particle fluorescence while under the observation. So we can look at conformational transitions, right? So if the molecule changed shapes, or if the molecule had interactions with other proteins, right? The fluctuations are going to be in the data, and that interaction will be in the data, and we can extract that data. All right, so there are many methods to produce a small volume. Typically, when we're using confocal microscopy, we're using the laser light source, right? So we have a laser light, we have a little to focus the beam onto this dichroic, and we're expanding the beam such that it goes through our objective. The objective focuses the beam to that small PSF right there. The emission will go back through this detector into the photo, let's say, in this case, some sort of photo counting detector, or it could be, you can also use cameras, but we can talk about camera FCS later. This is just for principle of single photons detection. We can also use 2 photon excitation, and we can, of course, 2 photon, multi-photon effects allows us to look at second harmonic generation, which we'll probably hear later in this course. And then, well, you can look at many other types of nonlinear interactions of, let's say, tissues. All right, so what does the focus spot looks like? With one photon excitation, the beam is going all the way through the sample, but the focus spot is right there in the center. We can eliminate the out of focus light. That comes back and we can generate the geometry of that focus beam. Two photon excitation, we have a high density, high probability of finding two photons in that particular spot to excite the electrons from our fluorescent molecules. That's exactly what we're doing, right? We're exciting electrons that are coming from our conjugated ring structure, as Professor Digman had showed, that the higher the number of double bonds, right, you're going to use either less energy or more energy to excite those molecules. Okay, so one thing to recall with two photon excitation is that the square dependence of light intensity, the probability of exciting the molecule goes with the square of the intensity of the laser beam. So if we increase the laser beam, it's not linear, right? It's not going to, my molecule, when it emits light, it's not going to be linear intensity. So if I double the intensity, it's not going to double my light doubling. The laser light is not going to double the intensity of the fluorophore. It's non-linear. It's going to be a quadratic effect. So it goes to the square of the intensity. So just to note that. All right, so let's look at the lateral size of the point spread function. We can characterize the lateral, let's say, diameter of the PSF. There are different ways to calculate the actual lateral resolution. This one is using, you can see in this particular equation, we have the full width half max, which is here. Others use another variable called 1 over e squared. So anyway, in this equation, we could see that the lateral resolution is proportional to two main things: the wavelength of light and the numerical aperture of the objective. So the numerical aperture of the objective is n sine theta. Where's that n sine theta coming from? It's actually coming from the objective lens. So how the lens is focusing, is it focusing at an angle, at a given angle? If it's focused like this, or if it's focused like this, the more it focuses, so the higher the numerical aperture of the lens, the better the point spread function, because it's proportional to the lateral resolution. So in this case, we're using the equation, the 1.22. Where is this coming from? It's coming from the Bessel function of the first zero, and the Bessel function arises from a Fourier transform of the aperture function. So that's where the 1.22 comes from. And we can distill this down to 0.61 lambda, which is the wavelength. So depending on whether we're using short wavelengths or longer wavelengths, and we can divide that by the numerical aperture. So shorter wavelengths will actually focus the beam much smaller, right? Because it's the longer the wavelengths, the beam waist is going to be much bigger. Okay. If we look at, because this is not just 2D, right? We're looking at a volume. We can look at the axial resolution of the point spread function, and the full width half max here is around 568 for this example. So in the axial resolution, we have 2n lambda, of course, is dependent on the wavelength divided by the numerical aperture squared. So if we have a 1.4 numerical aperture objective at 550 nanometers, our axial resolution will be 850 nanometers. Okay, so as a rule of thumb, when we talk about the volume of the point spread function for one photon excitation, we say that it's approximately 3 times the volume. When we're talking about 2 photon excitation, it's approximately 5 times the volume of the lateral resolution. So whatever the lateral resolution is, it'll be 5 times that in the beam. A little bit elongated with 2 photon excitation. Okay, so when we're using 2 photon lasers, we usually use a TiSAF laser, those titanium sapphires. lasers, the reason they're named that is because they're actually using that crystal to generate the lasing of the light. And it has a mode lock oscillator, which means that it has a given frequency, and the mode locking also enables you to have a tunable wavelength. So between 650 to 1,100 nanometer wavelengths, this is from the red to the infrared. And the pulses are ultra short. So that's the pulse duration is 10 to 1,000 femtosecond pulse duration, ultra short pulses, but at a given like 80 megahertz repetition rate. So short pulses means that you have high photon density, but low average energy. And we have high bandwidth, which means that we can tune the laser at any wavelength that we want. We're going to have a high probability of exciting those molecules. So this is just an applet. If you want, I can send you, well, I'll give you these slides so you can play with the applet and change the laser wavelength and using 1 photon and 2 photon excitation. So now that we talked about the laser and the point spread function, we're going to go to looking at the molecules. All right. So what do we see here? We have 2 different concentrations. We have low concentration and high concentration of molecules. And under the low concentration condition, very few molecules can transit the beam, right? In this case, I'm only showing 2 molecules, but that's fine. There's an experiment that was done a long time ago. Can we just detect 1 molecule? If we wait long enough and that molecule goes into our laser beam, then yes, we will be able to detect it. And that's good enough to generate statistics, right? Maybe, maybe too low is not good enough. But there was a principle stating that if you wait long enough and you see that molecule multiple times, then it's good enough to get a autocorrelation curve. We have many molecules. What will happen is 2 things. First of all, in this condition, you'll see the fluorescence intensity is low because we have few molecules. In this case, we have the fluorescence intensity is going to be much higher because we have many molecules. And you'll see that here the fluctuations are more, the amplitude is very high versus here the amplitudes are very, very low. Okay, so those 2 things. And the autocorrelation function here is the function where we're, let's say, calculating the analysis of the fluctuation from one point in time and saying, is this the same molecule in the next point in time? And if they autocorrelate, if you have a high probability of seeing the same molecule in a very short time scale, and as you go further in time, you will no longer see that molecule, right? So it's almost like the probability of seeing that same molecule in a short time scale, if it's diffusing very quickly, you're going to get this very sharp peak. But as it diffuses slower, you're going to get more correlation as a function of time, and then it eventually is going to decay. It's going to just go down. So what can we get from the autocorrelation function is the distribution of the amplitude of the function. I did not talk about that. What this means is that so you notice that the amplitude here is different than this amplitude in the sense that in the y-axis, when tau is equal to 0, so g of tau, and this is 0, the amplitude will be proportional to gamma divided by n. Gamma is the geometry of the volume of the point spread function, and it's a known value, so you don't have to calculate this. You can just use this value that's been calculated before, and divided by n, which is the number of molecules. So you'll see that the number of molecules is inversely proportional to the amplitude of the function. So if the amplitude is very high, that means you have few molecules. If the amplitude of the function goes down, that means you have many, many molecules, right? So it's inversely proportional to n, the number of molecules. So the amplitude, we get the amplitude and we get the distribution of the duration time of the fluctuations. So we need to extract this information to get the diffusion. We can also use the same fluctuations to get something called a calculation based on the distribution, what you saw in Schwetzberg's data, right? The frequency of the number of molecules that we see. Do we see 0? How many times? Do we see 1? How many times? And we can calculate the Poissonian distribution or the PCH distribution. So this is the photon counting histogram analysis from the same data set. Okay, so we have here fluorescence intensity as a function of time for a molecule that's entering our observation volume. And at some point in time, it's going to give off a photon. It's not going to be emitting the entire time, right? It's going to be emitting because we're pulsing, right? We have a pulse duration time, we have a frequency. So eventually a photon is going to be emitted at a given time. If the molecule stays in our volume, it's going to emit another photon, and it continuously diffuses, going to emit another photon. So that's going to happen very, very quickly and very fast. So by calculating the correlation at the intensity, right, if we're looking at intensity at time 1, we can multiply that by the intensity at time 1 plus tau. So the time delay at the intensity further in time, divided by the intensity average square to the square, and then we normalize to 1. And this correlation function enables us to now plot the graph of if the correlations we have many molecules, right? So we're just not correlating like one time point and then the next time point. We're doing like a shift to correlate each time point. And you'll see that at a very short time scale, we have a high probability of seeing that molecule. And as a function of time, the molecules will be gone. And we have good enough statistics that we generate a average correlation of the diffusion time of the molecule. So we can now take this curve, fit it to an equation, and tau, let's say at 1/2 of this correlation function, we get the time of transit, how long the molecule stayed in that observation volume as a function of time. We also can calculate the amplitude of the function. G at time 0, when tau is equal to 0, is proportional to gamma, and we say 1 over n, but it's actually gamma divided by n in that particular time. Okay, so just looking more in detail, what are we seeing is that we're correlating the fluctuation at this point in time with a delayed fluctuation as a function of time. And we do these correlations all the way across the graph, and it doesn't matter if we cut this. If we cut it into shorter time segments, it doesn't matter very much. Similar to Schwetzberg blinking and not getting the number of molecules at that time, right? It doesn't matter because statistically, we're going to get a correlation function for, we can't cut it too short because we need to get enough statistics. But if we have a long trace, we can cut it in multiple segments to generate autocorrelation functions for the statistics. Any questions so far? Okay. All right. Yeah. That is, so we'll know this value. You can see by eye 0.3 something. Yes. So you can fit this to get the G when tau is equal to 0, right? So when we do the fitting, you'll have a G of 0. And that, so because we don't, like at this point in time, it will be, we don't get any data, but we extrapolate the function to get this value. And you can do it by eye. I mean, I'm just looking 0.32 or something like this. That would be my G of 0. So that would be 0.3 is equal to, or let's say is equal to gamma divided by n. We solve for n. We know what gamma is. And that's how we get n. We get the number, and then we need to use Avogadro's number and let's say the volume of our point spread function, which is approximately a femtoliter. So we have femtoliter, we use Avogadro's number, because that's the number of molecules per mole. And then we can get moles per liter. That's how we get the concentration.
And that's independent of the fluorophore or whatever you're looking at?
Independent of the fluorophore.
So you know the amount of molecules that you have there independently of the medium that fluorophore is. So the quantum yield is independent of everything.
So let's say we purify GFP and we take the green fluorescent protein and we dilute it to 100 nanomolar. Yes?
Yeah.
And we know this through absorbance. We can correlate that with absorbance spectrophotometry. So we'll put a drop in our microscope. We'll measure at a single point, or we'll do the correlation of 100 nanomolar of GFP. And that amplitude here will give me the G of 0. I solve for n, and I'll get the number. And using Avogadro's number, I should get back the 100 nanomolar. So now,
yeah, but the intensity of the fluorescent is not, I don't know if it's going to be the same in the through absorbance, so what you can look at in a cuvette, or when you're doing like flap inside a molecule, for example.
So the only way that that number will change is if the photophysics, right? Let's say that the GFP is in a different pH, or something happened to the GFP that changed the fluorescence intensity of that molecule. Certainly, the G of 0 in that case will tell you that there was a change in the environment. There was some sort of confirmation, something happened to the fluorophore photophysics of that, so you don't get the same concentration.
Oh, okay.
Yeah. Yeah.
Thank you.
Yes. Okay. So if we look at the autocorrelation function, here is the function, and there are many factors that influence the fluorescence signal, the quantum yield, and the detector sensitivity. So quantum yield is the amount of photons that is put into the system and the amount of photons that are coming out of the system. Or let's say of the molecule. The way that quantum yield can change for a particular fluorophore is due to its photophysics, right? So if the environment changes, then the quantum yield can change. And I think Professor Dave Jamieson may have shown some examples of, let's say, adding, what was the most famous one? The temperature change of, yes, exactly. NADH is a perfect molecule to talk about quantum yield. NADH has a structure that has 2 rings at the end of it. When it's free, these 2 rings go like this. It like does this. So when the light hits it, it has a very low probability of exciting those electrons. So the intensity is very, very low. Now, when NADH binds to a protein, those rings open up. Now you have a high probability of exciting the electrons of that molecule. So the intensity of an NADH in the bound form is very, very high. Okay. And for FLIM, the quantum yield is important because the dimmer the molecule, the shorter the lifetime, the more bright the molecule, the longer the lifetime, generally. Okay, so quantum yield can affect, of course, detector sensitivity can also affect. The WR describes our observation volume, and then CRT is the concentration over time. So that's the function that it can affect. All right, so when we're looking at the autocorrelation, we said that, the G of tau is equal to 1 divided by n, and all of this other stuff. What are we looking at? N is proportional to the inverse of N is when G of tau is equal to 0. The T of D, which is the diffusion time, so we can measure that in milliseconds time scale. And then the X and Y of the volume of the point spread function also matters. So when we fit the autocorrelation function, we must determine the volume of the point spread function. This would be in the X direction and the Z direction. And we talked about how that is calculated. You can theoretically calculate it for your system, or you can measure. I think it's best to measure it for your system because you never know in that case. So if we have T of D here, we can solve for the diffusion. So we would know the waste, right? This is the point spread function, and now it's generally measured in nanometers. So let's say most of the volume of the excitation is around 400 below 400 nanometers. So that would be squared divided by 4 tau of D, which would be the half time of the decay, and then you would get the diffusion value. So this is what it's saying. So we can calculate, for example, rhodamine 6G has a diffusion of 426 micrometers squared per second. Rhodamine is a very, very small molecule. And one thing I have not talked about is the size of the molecule. If the size of the molecule is very, very large, it's going to diffuse slowly. If the size of the molecule is very, very small, it's going to diffuse much faster. And similar to the concentration, can be calculated for N. And we've already had, this is the simplest case. This is the 3D Gaussian excitation volume. So if we're fitting to a 3D Gaussian, or there's a 3D Gaussian Lorentzian, there's many types of fits depending on the type of laser you're using. If you're using 1 photon excitation, you're going to be using a Gaussian fitting calculation. If you're using 2 photon excitation, you will be using Gaussian Lorentzian to fit the autocorrelation function. All right, so let's go back to N. So I showed you already that the amplitude changes and they're inversely proportional. So few molecules, in this case there's 2, the higher the amplitude. More molecules, the amplitude starts to go down and decrease. And if we look at why G0 is proportional to the number of molecules, we'll know that that comes from the Poissonian distribution that describes the statistics of the particle occupation fluctuation of the Poissonian system. And we knew from Schwetzberg's data, it behaved as in a Poissonian behavior, and so do these molecules as they fluctuate as a function of time. So if we look at the variance of the fluctuations, it's equal to the number of particles. Right? So here we have the autocorrelation function, and we write the variance f of t. This is f of t represents the average or the mean value of f of t over time. That's fluctuations as a function of time. The variance then is equal to this equation here. And so when we calculate the g of 0, it's just variance divided by n squared, which is 1 over n. How does it affect the observation? If the intensity increases by a factor of 4. So you can easily increase the brightness of the molecule by increasing the laser power, right? We can go from very dim to very high intensity if we crank up the laser. So now if we're looking at the intensity as a function of time, in this calculation, we see, let's say the intensity is 1, then 0, 0, 0, the intensity goes to 2, then the intensity goes 1, 1. We calculate the average is 0.275. You can do this right now in your calculator. You can calculate the average of this number, 0.275. If we calculate the variance of that, of these values, we get 0.56. And now we already say that the average squared divided by the variance, we get n, 0.296. If we increase the intensity by 4, do we get the same n value? And that's the question. The average of all these values gives you 1.1. The variance of all these values gives you 4.09. N will be exactly the same. So we're not changing the number of molecules, we're simply increasing the intensity, right? By increasing the laser power, but number of molecules remains the same due to this fact that the variance is related to this Pisonian statistics. All right, so just to note what happens in that case. All right, let's look at different diffusion values. So I showed you rhodamine is around the hundreds, right? Like 400, 300 micrometers squared per second. That's a very, very small molecule, is going to diffuse very fast. So if you look here, the fast diffusion in the red, you can see the time here is shorter. Let's say if you measure GFP in solution, it's going to have a higher hydrodynamic radius. Recall that the hydrodynamic radius is related to the Stokes-Einstein equation, which describes here the diffusion time using Boltzmann's constant. We have here, and it's dependent on temperature, divided by 6 pi nu times R. And the hydrodynamic radius R here, the molecular weight or the volume goes to the power of 3. So if we have, let's say, GFP in solution, that's approximately 90 micrometers squared per second. You see the blue curve right here. We can certainly tell the difference between the red and the blue curve is very distinct. But now when you have very, very, let's say, small differences between diffusion, so now we do 70 micrometers squared per second, you see that the curve is not very far away from the blue. The green and blue are very close together. Right? So what this means is that if the molecule dimerizes, the autocorrelation function will shift very slightly. It's not going to be a huge shift. So between a monomer and a dimer, the change in diffusion is only a factor, goes to the 2 to the 1/3 or 1.26 increase, let's say, decrease in diffusion. That's not very much. Right, so if you ask me, can I tell the difference between a monomer and a dimer using FCS? The answer is not too much. No, because this shift is very, very small. The hydrodynamic radius should shift at least by a factor of, well, what do we say? Approximately, like some people say 8-fold increase of how big the molecule has to be distinguishable in its diffusion coefficients, right? There are other processes that can affect the autocorrelation function. This is the diffusion normal time of the molecule, let's say transit in the volume. But there's all of these other processes that can also take place. And that depends on how fast your data acquisition is. If you have a very fast data acquisition system, you can see photophysics such as anti-bunching. And generally, I have a definition of what these terms are, right? So you have molecules here. What is anti-bunching? This is the fastest process that can be recovered, and it's the probability for a fluorescence emission itself. So because we're trying to excite molecules, they're going to be anti-correlated at this very fast time point because you're exciting, and you can't excite so fast the next molecule. So there's going to be an anti-correlation here. And now when we go further in time, so if we increase the time, we are able to observe other processes such as rotational orientation of the molecule. So we can calculate rotation correlation functions that tells us how the molecule is rotating. So a rotational motion, this is like photo selection. And then there's another process called triplet state process. And this is the process of intersystem crossing. So if you remember Jablonski's diagram, there's a process called intersystems crossing to a triplet state, and that's like a blinking state. So for some period in time, the molecule turns off and then it turns back on. And these things are not really too well talked about in biological complexes. But if you're interested in, if you're ever purifying proteins, as Professor Dave Jamieson said, like tryptophan, looking at tryptophan, FCS, and things like that, these might be other states that might interest you in QVET studies. Okay, so let me show you an example of the correlation function, what happens in this example that was published in 2006 by this group. And basically they used DNA molecules that have a pharmaceutical potential in gene therapy. So folks are thinking maybe we can use DNA as genetic therapy. They are, however, large and negatively charged, right? DNA is negatively charged, and it's difficult transport over cellular membranes. So can we look at DNA compaction using polycationic molecules such as spermine? So these researchers used spermine, which compacts DNA, and this is in solution. So what happened is a function of time, and you have here the diffusion as they increase spermine, and we have on this side, we have particle number. And notice here the diffusion coefficient are those boxes here, which relates to this side of the axis. So as a function of concentration of spermine, the DNA is being condensed, the solid diffusion increase. So what is happening is the DNA is labeled, I don't know if I have a picture of this, let me see, yes. So the DNA here is labeled with some dye, pico green, and you'll see the DNA is very, very long. So the hydrodynamic radius of the DNA is big. And so what we're looking at is as the whole molecule here of DNA, right? I mean, this is kind of exaggerated. I don't think it's this big, but just to exaggerate the picture. We have this whole thing is diffusing as a unit. And as a function of concentration of this positively charged molecule, what happens is that the DNA compacts. So the hydrodynamic radius of this now gets smaller. And so the diffusion coefficient goes up. And what happens here is that the number of molecules goes down, right? So what's happening is you're going from like many molecules here, but to one molecule, observable one molecule here. Okay, so this is an experiment. So particle number decreases as the multiple DNA label becomes smaller and the detection volume becomes smaller. And this is important in gene therapy because we want to be able to deliver DNA into the cell. The idea is to try to compact the DNA to make it deliverable, right? So using diffusion. I don't know how much time I have. Like 1 minute. Okay. So we're almost done. So let's take a look at what happens in cells. So most of us are interested in cell work. And this is an experiment that was done in Rico's lab in Urbana-Champaign with a postdoc and student that became postdoc there. And she was doing this work on adenylate kinase tag GFP in HeLa cells. And there's different examples of HeLa cells. One was expressing adenylate kinase 1, and the other one's adenylate kinase 1 beta. And you could see that this protein is cytosolic, it's everywhere in the cell, and this protein here is localized to the plasma membrane. So if we look at the diffusion of GFP in solution, this is the yellow curve. It's very fast because it's closer to the fast time scale. And if we look at the diffusion of adenylate kinase in the cytosol, we see here that it is here, it's much slower than solution. It's even slower than GFP alone, right? So this is GFP alone, and this is in the cytosol, the diffusion, whether it's in adenylate kinase beta or not, the beta form. And if we look specifically in the membrane-bound diffusion, we can actually see almost two components. We have a fast component here, and we have a slower component here. So clearly there are two different species that dominate the correlation function of a very fast, right? This is the fast time scale and a slower diffusive component. Indeed, she did these experiments where she's looking at the spot at the membrane, and she got two diffusions using two species fit model. And one was a, let's say, fast diffusion of 10 micrometers squared per second. How much do you think it is in the cell? Anyone know the diffusion coefficient of GFP in the cell? Andrea, I know you know. Yeah. 20. It's around 20 micrometers squared per second, right? So so even in the cytosol, it's this adenylate kinase is slower than in GFP in the cell. And the membrane, you have a much slower diffusion component of 0.12. So definitely it's a bound protein and it's fluctuating still. Of course, there's steady state, right? We have proteins that bind and come off at the membrane, but we're only looking at the diffusion on the membrane at much slow calculations. So of course, this is really nice because you can take multiple FCS spots and then we can calculate the diffusion, but there's some challenges. We don't get an image from the measurement as just a point. There's some benefits. We have high temporal resolution, and even we can see rotational diffusion can be measured. And if we use something unique like STED FCS, we can actually use very, very small volumes. I'm going to give you an example really quickly on this. The challenge is that we can, immobile fractions can photo bleach, and the correlation curves can be difficult to interpret in regards to what's happening in the system. We can also do two-color experiments where we're looking at the green, let's say detector 1, and the red detector 2, and we can do a cross correlation to determine if the two proteins are bound together, right? And what happens is that a lot of biologists come to me and say, oh, but I can do colocalization. I can see if those two molecules are colocalized in a pixel. And I say, great. All right, let's take the situation. I have two molecules in the same pixel, but how do I know they're bound? They're like, oh, but they're in the same pixel. Like, yeah, like I'm sitting in a room and I'm sitting here, my colleague's there, we're in the same room, but that's it, right? Okay, how do we know that these two molecules are not like just holding hands, walking around together? We need to do this. We need to do cross correlation in order to figure out if the fluctuations are coincident with one another. And if they're bound together, they will cross correlate. So we can also do other things. This is the removal of the detector noise by cross correlation. So if you have noise in your detector and you do the cross, the noise shows up in the individual correlation functions in channel 1 and channel 2, let's say. But then when you do the cross, that just is gone. So you don't have to worry about the detector noise. And this is the cross correlation analysis where we have the intensity at this point in time. And then we're correlating how the cross works with the intensity as a function of time delay, but from channel 2. Right? And if they're coincident, if the molecules are like coming from the same species, statistically, they will cross correlate. The same species meaning they're both together, right? In the same time scale. And here's the cross correlation calculation that we use for fitting. And of course, we have to calculate the two color. We have the autocorrelation of one color, the autocorrelation from the second color, and we'll cross correlate. And what happens to the amplitude is the amplitude goes down. We have to ensure if we're using one photon excitation, the volumes overlap, right? You need to make sure that they're parfocal, like the two volumes are in the same spot for, let's say, your green laser and your red laser, and you don't want it to have like improper alignment of those functions. For two-color FCS, if they're uncorrelated, they're not bound. Of course, if they're correlated, they're bound together. And then, of course, there's other processes that we're not going to talk about called interconverting. And it's just, this is the function to calculate the intensities as seen in channel 1 and the fluctuations as seen in channel 2. If we have 100 molecules in channel 1 and 50 molecules in channel 2, then what will happen, as I mentioned, the cross correlation will go down, right? The fraction of, it's the fraction contribution of the channel 1 versus channel 2. So the fraction cannot be larger than the smallest of the G zeros, because we have 100 molecules, 50 molecules, and it cannot be higher. We can never go up in number of molecules, right? It will just go down. So that's why the amplitude goes down. Okay, so yep, I think I'm going to stop there.